Hello students, today we are going to learn molecular genetics part 6. In this part we are going to learn about DNA replication. DNA is a double-stranded structure. It is made out of two strands and these two strands forms the helical structure. They turn, they twist and form the helical structure. And each strand is made out of nucleotides. And the nitrogenous bases are thymine, adenine, cytosine, and guanine. Both the strands are complementary. Both the strands are complementary to each other. In the sense, wherever adenine is present, in the opposite strand, the thymine will be present. You can see wherever the adenine is present, in the opposite strand, the thymine will be present. And wherever the guanine is present, in the opposite strand, the cytosine will be present. That means the adenine will always pair up with thymine and cytosine will always pair up with guanine here. Both the strands will have opposite polarity. One strand will run in 5' to 3' direction. The other strand will run in 3' to 5' direction. And this polarity plays an important role in DNA replication. Now let's learn the enzymes involved in DNA replication. The DNA replication starts with the unwinding of double helical structure. And for unwinding, the enzyme helicase is required. And for the synthesis to initiate the process of replication, the enzyme called primase is needed. The primase function is the synthesis of RNA primer. And the DNA polymerase I, which is called as Quarenberg enzyme, it does repair mechanism. Even DNA polymerase II helps in repair mechanism of the DNA. And DNA polymerase III, it helps in the polymerization of nucleotides. It adds the nucleotides in 5' to 3' direction. And topoisomerase, which is also called as DNA gyrase, it prevents supercoiling of the DNA during uncoiling. And DNA ligase, it seals the gaps in the newly synthesized strands. These are the enzymes which are involved in DNA replication. Now let's see the actual process of replication. Now the first step in the mechanism of replication is the initiation of replication. The replication starts with unwinding of the DNA double helical structure. The unwinding of the DNA double helical structure. And the DNA replication starts at the initiation site and we call that site as ori of replication, ORI region we call it as. In prokaryotes, there is only one origin of replication. But coming to eukaryotes, there are several ORI regions. Since DNA strands cannot open at a stretch throughout, the replication fork will occur within the small openings of the DNA helix, and that opening we call it as replication fork here. And in the replication fork, so the replication to start, the unwinding of DNA should happen. So the unwinding of DNA happens with the help of the enzyme helicase. We call it as helicase. It is like unzipping. It is like unzipping of the DNA double helix by breaking the hydrogen bonds which are holding the both strands here. So helicase works in the same way. So the zip, if we take the zip, so it will open it. So to unzip it, we have to move in this direction. So here the unzipping will happen. If we consider this as one DNA strand and this as the another DNA strand and these metal as the hydrogen bonds, the unzipping is happening with the enzyme helicase. If we consider this as a helicase, this is how the opening of DNA helix happens. As a result of the opening of the DNA helix, the replication fork will be formed. This is called as replication fork. So this is how it looks like, the replication fork here. So the unwinding is carried out by the DNA helicase. At the same time, the topoisomerase enzyme will prevent supercoiling of the DNA during unwinding. So if you take a rope like this, and if you try to unwind at one end, at the another end you can see the supercoiling like this. In the same way, the DNA double helix, when it is getting uncoiled, the other side there is a chance of supercoiling. That supercoiling is prevented by this enzyme topoisomerase here. The step 2 in the DNA replication is the synthesis of RNA primer. RNA primer is a small stitch of RNA. You can see here, this is a small stitch of RNA here. And this is the starting point for the DNA synthesis, the synthesis of the new strands. So in the previous slide, we learned that the DNA helicase will unwind the double helical structure. As a result, the two separated strands here, this is one strand which got separated and this is the another strand. Both the strands will act like a template for the synthesis of the new strand here. And for the synthesis of the new strand, a starting point is required, and that starting point is the primer, which is synthesized by the enzyme primase. The primer is a small stitch of RNA. And why this primer is required for this replication here? Because the DNA polymerase, if you take the DNA polymerase, which we have seen already in the first slide, this acts in the polymerization. It helps in the polymerization of nucleotides. That means joining the nucleotides, adding the nucleotides here. It can add the nucleotides to the existing strand only. It can add the nucleotides to the existing strand. Suppose if RNA primer is there, it keeps on adding the nucleotides to the existing strand. And after replication, when the replication gets over, at the end, all these primers are removed. All these primers are removed before replication is complete here. Okay, this is about the RNA primer. Now let's learn the step 3 in the DNA replication, that is the elongation of the DNA strand. So for the elongation of the DNA strand, the addition of nucleotides should happen, and the enzyme DNA polymerase will keep on adding the nucleotides to the newly synthesized DNA strands. And here, to add the nucleotides, it needs the primer here. And one more condition for the DNA polymerase is it can add the nucleotides only in 5' to 3' direction. Now listen carefully. This is one strand which is acting as a template, and this is the another strand which is acting as a template. And this strand, this template strand is running in 3' to 5' direction. And the newly synthesized strand, which is getting synthesized with this template, will have the opposite polarity to this template strand. That means the newly synthesized strand will have 5' to 3' polarity here. Okay, and then if you take this template strand, its polarity is 5' to 3'. And the newly synthesized strand on this template will have the opposite polarity, that is 3' to 5'. This is the 3' end of the newly synthesized strand. This is the 5' end of the newly synthesized strand. So here they're having, these two strands are having opposite polarity, and these two strands are having opposite polarity, where one is the template, the other is the newly synthesized strand. Here also the same case. So the condition is the DNA polymerase can add the nucleotides only in the 5' to 3' direction. And if you take the DNA DNA synthesis will happen in two ways. One we call it as leading strand, the other we call it as lagging strand. Now let's see what is the leading strand. If you take in this strand here, what happens? Since the newly synthesized strand is running in 5' to 3' direction, there won't be any problem. The addition of nucleotides will be continuous. The strand will be continuous because its polarity is 5' to 3'. And we call that continuously synthesized strand as the DNA strand. So the strand, the one strand with the polarity 3' to 5', the strand which is having the polarity 3' to 5', the newly synthesized strand on that template will have the continuous, it will be continuous here, and we call that strand as the leading strand here. And coming to the opposite strand, the lagging strand, the another strand, we call this is the template and we call it as coding strand also. And where the DNA strand, the parent strand is having 5' to 3' polarity here, the replication is discontinuous. Why? Because here if you take, so here the opposite strand is having, the newly synthesized strand is having 3' to 5' polarity. And the addition of nucleotides is very difficult for the DNA here. For that, what will happen? A small RNA primer will be synthesized and then newly synthesized strands will be added. And next, new nucleotides will be added here. And again, a small stretch of RNA primer and then newly synthesized strands will be added here. So what happens? The synthesis of the DNA strand will be discontinuous. The replication is discontinuous on the other strand, okay, on the strand which is having the polarity 5' to 3'. And this discontinuously synthesized fragments, we call them as Okazaki fragments here. And the one condition why this strand is discontinuous, because it is running in 3' to 5' direction. That is one. And the second is the nucleotides are added. The elongation of strand is happening against the force of uncoiling. So here the force, the uncoiling is happening in this direction. And the addition of nucleotides is happening in opposite direction in this strand. So because of that force, what is happening? The strand, the synthesis of the newly synthesized strand, the addition of the nucleotides will be discontinuous in this strand. So this strand is called as lagging strand. And the fragments, the discontinuously synthesized fragments are called Okazaki fragments. And this strand is called as leading strand. And in the final step, when the addition of nucleotide is done by the DNA polymerase 3, now the DNA polymerase 1, the exonuclease activity of the DNA polymerase 1 will remove the RNA primer. So the RNA primer should be removed here. And after removing, the gaps will be there. So you can say this is the RNA primer. If you take the leading strand, this is the RNA primer, and here the continuous strand of the DNA. If you take the lagging strand, we have the primer and the DNA primer and the newly synthesized DNA, like that it will be there. So it is discontinuous. Then what happens? The primers are removed by the exonuclease activity of the DNA polymerase, and the gaps which are formed after removing, the gaps will be formed, and those gaps are sealed off by the DNA ligase. We call the DNA ligase as a sealing enzyme. It seals the gaps. You can see here the gaps. So this is the DNA ligase at the site, and it attaches, as a result, the gap will be sealed. So this is about the DNA replication.